Hello everyone, and welcome back to All Things GC. In today's video, we're going to be discussing epigenetics. Epigenetics refers to heritable changes in gene expression that occur without altering the DNA sequence itself. This distinction is important. When we traditionally think about genetics, we often focus on variants within the DNA sequence. For example, a pathogenic variant may alter a protein structure or function and result in disease. Epigenetics is different. The DNA sequence itself remains unchanged. Instead, mechanisms exist that regulate whether genes are expressed, how strongly they are expressed, and when they are expressed. As a result, two cells can contain the same DNA sequence, but exhibit dramatically different patterns of gene expression. This is one of the reasons that neurons, hepatocytes, skeletal muscle cells, and skin cells can all perform different functions, despite containing essentially identical genomic information. So why is epigenetics necessary? Every nucleated cell in the body contains approximately the same genome. If every gene were expressed equally in every cell, cellular specialization would be impossible. Instead, cells selectively activate genes required for their specific function, while suppressing genes that are unnecessary. This process is known as differential gene expression. Epigenetic mechanisms help establish and maintain these expression patterns throughout development and adulthood. For example, a neuron requires expression of genes involved in neurotransmission. A hepatocyte requires expression of genes involved in metabolism and detoxification. And a cardiac myocyte requires expression of genes involved in contraction. Although the DNA sequence is the same, the epigenetic landscape differs substantially. Now, the most well-known epigenetic mechanism is DNA methylation. DNA methylation involves the addition of a methyl group to cytosine residues, most commonly within CpG dinucleotides. Before discussing the effect of methylation, let's first review what a promoter is. A promoter is a regulatory region located near the beginning of a gene that helps recruit transcriptional machinery. When transcription factors and RNA polymerase can access the promoter, transcription is more likely to occur. When promoter regions become heavily methylated, transcription is often reduced. In simplified terms, increased promoter methylation generally leads to decreased gene expression. This is probably the single most important association to remember when first learning epigenetics. However, it's important to recognize that the relationship between methylation and expression is not always straightforward and depends on genomic context. DNA methylation can reduce gene expression through several mechanisms. First, methyl groups may physically interfere with transcription factor binding. Second, methylated DNA recruits proteins that promote chromatin condensation. This creates a more compact chromatin structure that becomes less accessible to transcriptional machinery. As accessibility decreases, transcription decreases. The gene remains present within the genome, but it becomes more difficult for the cell to utilize it. To understand the next mechanism, we need to briefly discuss chromatin. Human DNA is approximately 2 meters long when stretched end to end. Obviously, that amount of DNA cannot fit into a microscopic nucleus without extensive packaging. DNA is therefore wrapped around proteins called histones. You can think of histones as spools and DNA as thread wrapped around those spools. The combination of DNA and associated proteins is called chromatin. Chromatin exists on a spectrum ranging from relatively open to highly condensed. Open chromatin is generally accessible for transcription. Condensed chromatin is generally transcriptionally inactive. Now let's take a look at histone modification. Histones possess protruding regions called histone tails. These tails can undergo various chemical modifications, including acetylation, methylation, phosphorylation, and ubiquitination. These modifications influence how tightly DNA is packaged around histones. One modification that students commonly learn first is histone acetylation. Histone acetylation is generally associated with a more open chromatin structure and increased transcription. Conversely, removal of acetyl groups often results in chromatin condensation and reduced transcription. Rather than changing the DNA sequence itself, histone modifications alter accessibility to that sequence. Another important epigenetic mechanism involves non-coding RNA. Not all RNA molecules function as templates for protein synthesis. Many RNA molecules function primarily in gene regulation. Examples include microRNAs, long non-coding RNAs, and small interfering RNAs. These molecules can influence transcription, RNA stability, and protein production. Although the mechanisms are complex, the major concept is that RNA molecules can participate directly in regulating gene expression. One of the most clinically relevant epigenetic phenomena is genomic imprinting. For most genes, both maternal and paternal copies have the potential to be expressed. Yet imprinted genes behave differently. In an imprinted region, one parental copy is epigenetically silenced. As a result, expression occurs primarily from either the maternal allele or the paternal allele. Importantly, which allele is active depends on parental origin. This is known as a parent of origin effect. And this is where epigenetics intersects directly with clinical genetics. Now, the classic example involves chromosome 15q11 to q13. This region contains genes that are differentially expressed depending on whether they are inherited from the mother or the father. If paternal gene expression is lost, the result may be prader-willi syndrome. If maternal UBE3A expression is lost, the result may be Angelman syndrome. These disorders illustrate that inheritance is not always simply about which DNA sequence is present. Sometimes it matters which parent contributed to that sequence. Another concept frequently linked to imprinting is uniparental disomy, or UPD. Normally, one chromosome is inherited from each parent. In UPD, both copies of a chromosome originate from the same parent. If that chromosome contains imprinted genes, abnormal expression patterns can occur. This is one mechanism that can produce disorders such as Prader-Willi syndrome and Angelman syndrome. Epigenetics also plays a major role in cancer biology. Many cancers demonstrate abnormal methylation patterns. Tumor suppressor genes may become hypermethylated and silenced. Conversely, genomic instability may result from widespread disruption of normal epigenetic regulation. Because epigenetic changes can sometimes be modified therapeutically, epigenetics has become an important target for cancer treatment research. To summarize, epigenetics refers to changes in gene expression that occur without changing the DNA sequence itself. The major epigenetic mechanisms include DNA methylation, histone modification, and non-coding RNA regulation. These mechanisms help regulate gene expression, cellular differentiation, genomic imprinting, and numerous disease processes. Understanding epigenetics is essential for understanding modern genetics because DNA sequence alone does not fully explain how genes are regulated and expressed. And that's your review of epigenetics. Hit that like and subscribe button for more quick study tips, and I'll see you next time.